All right, so we're continuing some chapters that we definitely covered in Bio 5 or previous classes, but we're going to get a little bit deeper into it, talking about the cell. So this entire chapter is going to just kind of sum up the cell and its structures, DNA, how DNA gets expressed, how DNA replicates, and then how cells replicate. So we're covering a lot here, but hopefully at least the general concepts for this particular chapter, this particular section of the class, feel familiar. So what are we going to cover in this particular video? We're going to look at the plasma membrane, see how the membrane works as a semi-permeable membrane. We'll look at cytosol versus cytoplasm. We'll talk about the cytoskeleton, different appendages that the cells can have, and then the organelles. So it's important to note we are going to focus on the eukaryotic cell since we are looking at human physiology and human cells are eukaryotic. And so there's really three main parts that all eukaryotic cells are going to have. So these three main parts are the plasma membrane, which is just creating that barrier between the inside and outside. However, it's not just a barrier like a wall, it's dynamic and is selective in terms of what comes through. The cytoplasm is a generic term we use to kind of talk about all the insides of the cell. But we can break the cytoplasm down into two parts. The cytosol, which is like the fluid, and the organelles, which are the components, right? The functioning components in the cell. Some of these organelles are bound by membranes, others are not. And then we get to the nucleus, which has our genetic material. So no matter what eukaryotic cell we're looking at, we're going to see these main components. Now, the number of certain organelles or what specific processes they're doing, that'll be different depending on the specific cell type. But no matter what cell we're looking at these components. And so here's our generic animal cell, basically, just kind of models all of these components. So this doesn't really look like any individual cell we would find in the human. It's just one that shows all these components together. And so we see the nucleus here, this big kind of yolk looking one. We see all the various organelles, including members of the endomembrane system, mitochondria, right, lysosomes, the centrosome. We see the cilia and flagella that are used for movement, as well as microvilli. So all sorts of different components that make up the cell. And we'll get to talking about each one of them as we go through the video. So we start with the plasma membrane. The plasma membrane is flexible yet sturdy. So one of the ways we like to describe this is it's the fluid mosaic model. Similar to like how a large crowd kind of moves as a unit, the plasma membrane is kind of similar in that aspect. So you have densely packed together phospholipids, glycoproteins, glycolipids, right, so on and so forth. But they can still move, right? So while the crowd is really hard to get through, individual people within the crowd can move around. And so we can kind of think of the plasma membrane in that same fashion, that it is a very crowded, packed together structure, right, with all these phospholipids and different proteins and proteins accessories all together. Each individual component can move around within the crowd, but overall the crowd is hard to get through. So it's semi-permeable. The membrane will have some regulatory powers over what comes in and out of the cell. We arrange this in a bilayer, basically a sandwich. So if we look at the phospholipids, we have the phosphate heads shown in that light blue, and the fatty acid tails shown as those little white matchsticks. If we're looking at it that way, the phospholipid heads on the very inside and very outside are water-loving, they're hydrophilic, while the fatty acid tails are hydrophobic. So because of that hydrophobic nature of the phospholipids, they're going to automatically arrange themselves in a way so the hydrophilic side faces water and the hydrophobic sides don't, right? So we see that's the arrangement. This arrangement will happen, right, without really any intervention. Right, this arrangement will just simply happen because of the chemistry of the phospholipids and the cytosol inside the cell, as well as the fluid surrounding the outside of the cell. And so we have various components, right? Glycoproteins, carbohydrates, proteins, glycolipids, cholesterol, different proteins that span the entire length of the membrane, some that are just embedded on one side. There's all sorts of different things we're going to get into with this. We won't get into all of them though, because I mean, we could spend forever talking about all the different components of the membrane. So remember I said the phospholipid bilayer, it follows what's called the fluid mosaic model, right? The fluid mosaic model. Why am I saying it over and over again? Because I will expect you to recognize fluid mosaic model and what that means, right? What this means is that this membrane it is not a solid structure. Like a fluid, it moves around, right? There's movement, it's dynamic. And so because it's dynamic, right, we can change, right? We can add things in, we can pull things out, right? So we have movement. These phospholipids have the ability to move around. So let's say we wanted to demonstrate this. We could tag some of these phospholipids with something that produces a fluorescent light, right? So if we shine a black light, it'll glow. We could tag a small portion of these all together in a little pile. And then if we were allowing the cell to just kind of do its thing, we would notice over time that all of the phospholipids we tagged with the glowing fluorescent protein would move around to the point where they would all be super dispersed out. They would not necessarily be next to each other. Right? That's helpful. We don't want to be a brick wall. We want some fluid, we want fluidity, we want some movement, because we are going to have to interact with the environment around us as well as the inside of the cell. Now that being said, we don't want to be too fluid so that we fall apart. And so we do have some components that help create a more stable, right, stable bilayer, namely cholesterol. So cholesterol, right, being one of those lipids, will be embedded within the bilayer as well, and that's going to help prevent it from getting too fluid-like. So we have the hydrophilic sides facing the very outside, very inside, and in the middle we have the hydrophobic center. The hydrophobic center, made of those fatty acid tails, really is the reason why this membrane is selectively permeable. Because it's a lipid-based center, things that are non-polar and small may be able to get through, but anything that is polar, anything that has a charge, anything that's really large, is not going to have an easy time getting through this. So we can use this lipid-based hydrophobic center to put restrictions on what's able to get through. And then we can use other components like a channel protein to allow specific things to get through. Maybe we even open them like it's an ion gated channel where we only allow it when we reach a certain threshold, when we get to a certain point. So if you are hydrophilic, right, you're polar, you're an ion, you're going to need a protein channel to get through this membrane. And we'll have plenty of those for when the time is right. Now, some of these proteins are integral or transmembrane proteins, meaning they go across the entire membrane. So we see one here kind of in the middle, all right, this guy right here that I'm circling with my mouse. And so we see it goes all the way from one side to the other, right? And it goes all the way through from one side to the other. So it has an impact on both the inside and outside. And then peripheral proteins, these are found on only one side. So we see like one right here that's only on the outside. And then this one right here, not the curlicue, right? This one right here is found just on the inside. So peripheral are found on the side, whether that's inside, outside, doesn't matter. Integral or transmembrane go all the way through. Now, why do we have these membrane proteins? Because again, remember, we mentioned that a lot of things cannot get through the lipid bilayer. We need some sort of channel. We need a protein to help us. Some of those channels are used for moving ions. So something like sodium can get through using an ion channel. Some are carriers. So we're kind of similar to the ion channel. The difference here is that we're going to actually kind of bind into the carrier protein and then change shape in order to get through. We can be a receptor. So basically, we will receive a signal via a ligand. A ligand is just like a signal, so it could be something like, let's say, a hormone. And that's going to then cause a change or cause an action inside of the cell. We have enzymes. These are integral and peripheral, that they're going to help with the cell's ability to speed up chemical reactions. We have linkers. These are going to basically just help with structure, stability, shape, right, of the cell. And then cell identity markers. This is important in your own immune system. So basically, like, this is going to mark your cells as self, right? So that your immune system, your body knows that that particular cell is supposed to be there. It's part of your body. Now, continuing on with the plasma membrane, we can have certain little appendages, little extras that can help us with certain specific functions. Most of the time we're talking about movement or mobility. So let's look at the first one, which is cilia. Cilia are small, short, hair-like structures that come from the plasma membrane. So they're like little extensions of it. And they're motile, so meaning they have movement. And really, these cilia, they're going to function together. So they're all going to beat together in the same direction, kind of like forming like little waves. We see these in the lining of your bronchi, in the lining of your trachea, your fallopian tubes, right? So they can be used to move the cell. They can also be used to move things around the cell. And so these are going to use microtubules in this, what we call a 9+2 arrangement, meaning there's 2 tubules in the center and then 9 pairs going around. So we have this arrangement. And then the cilium, that base, is going to have a pair of centrioles, which forms what's called the basal body to control movement. So the centrioles help control the cilia. If you're trying to think of what to remember for all this, the biggest thing is knowing what cilia are for, their basic structure in terms of what they look like, and knowing that they are composed of these microtubules controlled by the centriole. Now, if we need to do some real good movement, a flagellum is great. Or flagella if we're talking about more than one, but usually we're talking about maybe 1 or 2 of these on a single cell. This is just one long structure like a tail that is going to be used to help move the cell. So it is motile, so it's going to cause movement. In this case, it's going to be a fast whip-like motion. And one of the best examples, really the only example in the human body of a cell that has this, is sperm. They also have that same microtubule 9+2 arrangement, very similar, and also controlled by centrioles. And then lastly, we get these microvilli. Now these are short, non-motile, so we are not moving, right? They're kind of like the cilia, except they are not worried about moving. These are just to increase surface area. So by having all these little projections that go up and back down, we're increasing surface area, and by increasing the surface area, we can basically create more space for chemical reactions to happen. Or let's say we're talking about the small intestine. Small intestine has these microvilli, and that's so that we can absorb as much as possible, right? If we increase the surface area, more of the nutrients from the food we eat can actually physically contact the cells, and if they physically contact the cells, we can absorb them in. We get to cytoplasm versus cytosol. Again, this one's pretty straightforward. Just cytoplasm is everything inside the cell. Cytosol is just the fluid, right? It has a semi-fluid consistency, so it's like a little bit thicker than like a water kind of thing, even though it does have a lot of water in it. We're looking at 75 to 90% water. Ions, proteins, organic molecules, but still going to be a little bit thicker. And this is going to be where a lot of different reactions occur. They're going to occur in that cytosol. Now, we have to have some shape to our cell, and some of the components that are going to help with that we've already discussed. So we've talked about different microtubules and whatnot. That's going to be one part, as well as intermediate filaments and microfilaments, and together the organized system of these three is called the cytoskeleton. So this is going to be what helps give us shape. It's going to give us structure. Sometimes it can even help us move. It can help us with anchoring to things, so we can anchor to things outside of the cell. And then lastly, we'll use this when it comes to cell division. So huge job that the cytoskeleton takes on, right? And so we basically see like a web, right, within the cell. And so this is going to help with all sorts of stuff. Like I said, it's going to help with the cell dividing. It's going to give shape, structure. It's also going to, in a way, serve as like our highway system for moving things around. So we have some cool proteins called motor proteins that will actually be able to use these microfilaments, use these microtubules to basically go around the cell like they're walking along a street. So here's the three basic types. We have the microfilaments, the intermediate, and the microtubules. Don't confuse microfilament and microtubule. Microfilaments, really thin. Think like a little tiny thread, right? Versus a microtubule is more like a big rope, right, with a hollow inside. So actin filament would be a good example, like actin would be a good example of the microfilament. Intermediate, things like keratin, and then things like alpha and beta tubulins would be a good example of the microtubules. Lastly, we get to the organelles. The organelles are just like the little organs found inside the cytosol. These are just basically specialized structures that perform a specific function. So the cell has done a very good job at compartmentalizing all the different things it needs to do. And so most of these jobs are done by one specific organelle. Some of these are bound by a membrane, others are not. So let's start with the ones that do not have membranes, like the centrioles. The centrioles are going to have this 9 and 3 configuration of microtubules. And so the centrioles are kind of the organizing center for a lot of the microtubules, a lot of the filaments that we see making up the cytoskeleton. So they're going to help with mitosis, forming the spindles so that we can move chromosomes around. It's also going to help anchor flagella and cilia down. The proteasome is going to be like this barrel-like structure that is basically when a protein's no longer needed or a protein's been damaged or isn't working right, a proteasome is going to basically break that protein down into smaller pieces so we can recycle those amino acids. So this, honestly, I think this is one of the cool ones because you think about all the proteins you're making on a daily basis. What happens when you don't need that protein anymore? What happens when you've already done the function and you need to stop that process? We have a way of turning that off. But I think the coolest thing with the proteasomes is the fact that we're not just destroying a protein completely, we're breaking down into amino acids that we can use to build a totally different protein. And that's cool, right? We're efficient, we're recycling, we're green, right? In terms of being able to reuse supplies so we don't have to bring in new amino acids all the time. Now, in order for the proteasome to work, we have ubiquitin, which is basically this marker that is going to mark the protein for degradation. So the proteasome has to find that ubiquitin chain before it can just destroy a protein. So we have some organization or some regulation here so that we're not just destroying any and all proteins that the proteasome comes across. We're only going to destroy the proteins that are marked for destruction. Next, we get to the ribosome. The ribosome is going to be our protein-making factories, right? They're responsible for protein synthesis. They can be found just free-floating on their own, or they can be found as part of the rough ER. They have two components to them, the rRNA, so ribosomal RNA, and protein. which is why we call it a riboprotein. And there's two subunits, a small and a large subunit. When we use the ribosome to produce proteins, the large and the small subunit are going to come together around the mRNA that is used as the template. So now we get to the membrane-bound ones, and we're going to start with the first of our few components that make up the endomembrane system. We're going to start with the endoplasmic reticulum. Because it's a bit of a mouthful, I'm going to call it the ER for short. So just know if I'm saying ER, I'm talking about the endoplasmic reticulum. This is going to be a network of membrane-bound sacs and tubes that are connected directly to the nuclear envelope. And there's two main types, the rough ER and the smooth ER. They're really honestly named because of how they appear when you look at them under a microscope. The rough ER appears rough, the smooth ER appears smooth. Scientists were not all that creative sometimes. But the reason why the rough ER looks rough is because ribosomes are actually studded into it. So when we see that rough appearance, so we're seeing the bumpiness that's created by the ribosomes sticking in the rough ER's membrane. So the rough ER is going to have a role in protein synthesis and modification, and the smooth ER, which does not have ribosomes, is going to play a bigger role in terms of lipid synthesis. The smooth ER can also be modified for specific functions when we look at specific cell types. So for example, if we look at muscle, we'll see that the smooth ER serves as a calcium ion reservoir when it comes to being able to organize and regulate muscle contraction. Lastly, we get to the Golgi complex. This is kind of called the mailing system, the UPS, the FedEx, the Amazon fulfillment center of the cell. Because basically what the Golgi complex or the Golgi apparatus is responsible for is sorting, packaging, and modifying things that need to be sent through the cell. So if you think of it, like FedEx doesn't make the things you order, Amazon doesn't necessarily make the things you order, but they do package them together in a box for you and send it to your house. And so that's what the Golgi apparatus is doing. So this is going to be made of these stacks of membranous sacs called cisternae. And so you see how they're kind of almost like a stack of pancakes or crepes is kind of how they look. That's how I would describe it. Everything's food with me. But we have those flattened stacks, and there is a cis side and there is a trans side. Things are going to arrive at the cis side or the cis face. As they go through the stacks, they'll then get to the end on the trans face where they can exit and be shipped out. So we look at how these things are kind of interacting together, right? We have some ribosomes in the rough ER, right, working together to synthesize proteins. The rough ER creates a vesicle with the protein in it. It fuses with the cis face of the Golgi complex. We then go through package modify as we go through, and then we get to the end of the Golgi complex or Golgi apparatus on the trans side, and we can create a new vesicle after being modified. From there, we can either move somewhere else within the cell. We can be a secretory vesicle where we're going to basically push these components out of the cell. We can transport something to like a lysosome or to another organelle, or we can merge with we can bring things out of the cell. So basically just push things out. Maybe we want to put them in the membrane. We want to embed them in the membrane itself. Something that comes from that Golgi complex is the lysosome. So that is one thing that the Golgi complex does make. And this is a special membranous sac, little kind of vesicle-like structure that has digestive enzymes. I like to think of the lysosome as the recycling facility of the cell because it can take bigger worn out components, like if there's an old mitochondrion, or if we bring in a large particle, like let's say we're a white blood cell and we just brought in a bacterial cell that was causing an infection, the lysosomes can then break those things down. It can break down the old mitochondrion, it can break down the bacterial cell we brought in, right? And then we can use the components that we broke down, we can reuse them again. Next, we get to the peroxisomes. These are going to be involved with detoxing harmful substances and helping with lipid synthesis, like making cholesterol. So we see these guys in the liver in high numbers, which if you think about it makes sense, right? Because you think your liver tends to filter things out, tends to break things down that need to be broken down. So peroxisomes being in high numbers in the liver sounds about right. So they're going to have enzymes that are oxidative. They're going to oxidize things. And again, just make sure that certain substances that tend to be toxic are broken down and left in a safe manner, safe form. Next, we get to the mitochondria. Mitochondria is going to be plural. Mitochondrion is singular. But you probably already know this as the powerhouse of the cell. But we want to get deeper than that. It's also going to be where cellular respiration is going to occur. And so basically we're going to be producing most of the ATP that our cells need using these. Now these are double membraned, so they have both an inner and an outer membrane. That's different from the other organelles we've seen. And the reason that we see that is because the thought is that mitochondria were actually free living. And so when our old ancient cells brought the mitochondria in, they still had their membrane and then got enveloped in a piece of the membrane of the ancient cell. The other evidence we have of this is that mitochondria have their own DNA. So also different, also very unique compared to other organelles, that they have their own DNA. We only inherit this from our mothers though, mainly because the mitochondria you have are coming from your mom. They're coming from the egg. When we have a cell that is metabolically active, like super active, so things like muscle, kidney cells that do a lot of work, we're going to see more mitochondria. Compared to cells that don't do this, that are not metabolically as active, so something like a red blood cell would not have very many, if any, mitochondria at all. Next, we get to the nucleus. The nucleus is going to house DNA. DNA is going to be stored in a form called chromatin, which is basically the DNA plus associated proteins that are going to help organize it. And the nucleus has a couple components to it. We have a nuclear envelope, which is a double membrane. And within this nuclear envelope, we have nuclear pores, because we're going to have things that need to get in and out. And then the nucleolus, which is that really dense, dark center in the middle there, is going to be where we make our RNA, basically where we're going to make one of the main components of ribosomes.